Thank you, Dave, for the introduction. Like I said, my name is Sarah Braun, and I'm a PhD candidate in Dr. Shelley Jansky's lab at the University of Wisconsin-Madison. And my talk is a case study, what to do when your SNP data set arrives. So I have three different parts that I'd like to go over for the presentation. One is what you can do before your data set arrives. Two is examples of Excel tools with phenotypic data. And I just want to note that this is in Microsoft 2010. Part three are some step-by-step exercises to prepare your SNP data for mapping. So part one, before your SNP data arrive, you can start by determining your mapping software. Are you going to use JoinMap, R, or another type? In this case, you may want a RAM of 6 or 8. The next thing to do is to organize files. If you already do not have a system, it's a good time to get organized with that. What I like to do is name files by dates and then track how many SNPs are relevant to each step and naming these tabs in Excel. And I'll go through this at the end. And some people in our lab are using a free and secure online storage space called Dropbox. I'm sure that there are others out there. But this is quite easy to download, and the benefit to this is that you can invite others to a folder where you can share files, edit files, things like that. So part two, some examples of Excel tools, and again, just to reiterate, I'll start with this section with phenotypic data. And first I'll start with the filter function. And the objective of the filter function is that we only want to see certain parts of our spreadsheet. So for this spreadsheet, in A I have rep, B is genotype, and C is response. So this is a phenotypic data set, and we only want to see 4.1 and 4.5. So the first thing we do is highlight row 1, and then, oops, click on it. And then click on data, and then click on the filter icon. And then these drop-down options will appear in row 1. So the next thing we want to do is click on the drop-down for the genotype column, and this box containing options for that column will appear. And then you just start clicking on boxes next to the genotypes that you do not want to see, and this deselects them. So you can see I've deselected 4.2, 4.3, 4.6, and then 4.7. So once you've removed the check marks from all the genotypes you do not wish to see, you click OK. So click down there. And then we only see 4.1 and 4.5. So there are a couple things I wanted to note about this is that one, we'll use this for the genotype data, so you can simply copy and paste this information into a new sheet. So if you only want to work with those, you now filter for them and you can copy and paste those into a new tab or a new worksheet. The second thing is that we can notice how many records we have when we set this type of filter. All right, another example of an Excel tool with phenotypic data is the CONCATENATE function. And so this is the same spreadsheet we were looking at before, but we want to, the objective is to concatenate rep and genotype, so we have a unique identifier for each record. So start by typing in equals concatenate, and then you click on B2, or you type in B2 to reference rep, a comma, and a dash, and then a comma, and then C2 to reference genotype, and then push enter. And then this gives us what we've asked for in the formula in this cell, and you fill in all the way down. So the one thing you want to remember when you're writing filters, or I'm sorry, when you're writing some commands in Excel, and this will be important when it comes to your genotypic data as well, is that you want to copy and paste special at each of these steps. So I'll go through how to copy and paste special really quickly. So you highlight your column and you click on copy, and then you go down to paste special, and then it defaults to all, which copies all of your formulas and everything, and you want to go down to values and click OK. So the next function I wanted to teach you guys was the VLOOKUP function. And the objective of this is that you have your master spreadsheet, so this is the one that we were just working on, and we've collected a second set of data. And the second set of data is in the supplemental spreadsheet. And this code in A of the supplemental spreadsheet is the same as the code in the master spreadsheet, so we'll use those two to match up. And response to is what we want into the master spreadsheet. So I'll start by highlighting and typing in where I want response to occur. So then in the master spreadsheet, I type in equals VLOOKUP, and then I highlight column A of this master spreadsheet. This is what the lookup value is that it asks you for in VLOOKUP. And then you highlight column A and B of the supplemental spreadsheet, and this is the table array. So there's one trick to remember about this, actually two, and that's that these two column A's need to be in the leftmost column. It's the easiest way to remember that. Otherwise, it won't know where to grab it from, and you'll get an error message. And the second thing is, before you even start this process, you probably want to sort by each of them, sort each of the codes so that it's in a similar order. Those two things can cause some error messages that you get. So anyway, we've highlighted what we want to grab from the supplemental spreadsheet. And next we type in 2, because response to is in the second column of the spreadsheet. So 1, 2. And then finally you type in false, because that gives you an exact match. So it's matching up, again, column A from the master and column A from the supplemental, and we want an exact match. So that's why it's really important to type in false, and as opposed to true, which will just give you an approximate match. So then you just fill in this formula all the way down, and you want to copy and paste special at the end of that one. The next function I wanted to show you guys is the true-false function. And for this one, we have the master spreadsheet and the supplemental spreadsheet, and I've just copied and pasted this information into the master spreadsheet. And my objective here is to see if column A and column E match in the master spreadsheet. So here we are in the master spreadsheet, and you type in equals A2, and then another equal sign, and then equals E2. And that will either tell you a false or true, and you can see for false that column A and column E do not match for row 2, and true, it's a true for row 3, because A3 and E3 match. So that will come in handy later. Another trick I wanted to show you guys with your phenotypic data is a pivot table function. And the objective of this is to summarize phenotypic data. Of course, this is just one of many ways to do this, but it's one way I like to summarize. So what I start by doing is going into Insert, Pivot Table, and then this box shows up and it asks you to select a table or a range. So you click on this to highlight the table range, and then you highlight everything. And then I like to say New Worksheet just to get the data separated out from what I'm working on, and click OK. So this is what your pivot table tab will look like when you initially get it. You see here down is row labels, and we'll go over this again, and here is values. Here are what your different columns are labeled. So we're looking at rep, genotype, response, and response to as your options. So first I'll click on genotype up here, and you'll see this appears as row labels, so 4.1 through 4.7, and it's appearing as a box here down under row labels. And then I click on rep, and this is under row labels as well, and you'll see that this falls under the each genotype that it belongs to. So if you have something like rep that shows up under values. And pivot tables are easiest once you start working with it yourselves. Know that if rep is over here, all you have to do is click on the box and drag it over here, and you'll see it correspond to go underneath here. Okay, and then we want to see response. We click on response, and this will appear under values. And so we can see the sum of response is under each of these. So we have 4, 1, reps 1 through 3, their respective values, and then a sum of those. So a lot of times we want to see averages and not sums. So you go down to the sum of response, which is in the bottom right-hand corner, and you click on value field settings. And in the value field settings, it actually defaults to sum, but you can change it to average. And then click OK. And then this will give you the average of what it finds for 4-1. Okay, so the next part we'll move on to working with SNP data. And this is where we are in the entire context of the entire process. First, you plant your population, you phenotype, and you collect tissue. 2, extract DNA and ship it for the SNP assay. Step 3, SNPs are read using Genome Studio software. Step 4 is really where we are, is where you receive your SNP data, and this is in a text file format. Next, you create your map where you develop relationship matrices and finally use for QTL analyses or other type of analyses that you're using. So this is a summary of the outline of the steps, which I will go through with you guys. The first one is moving the text file to Excel. 2 is adding the annotation. And when we add this annotation, you can either use the true-false function or the VLOOKUP function to make sure that everything's lining up. And in our case, we'll use the true-false function. Step 3 is to filter out SNPs missing data from either parent. Step 4, filtering out bad and questionable SNPs. Step 5, filtering out greater than 2 hits, which is 2 SNPs on the same pseudomolecule. Step 6 is filtering out SNPs with missing data for greater than 10 progeny. And finally, separating the heterozygous by homozygous, homozygous by heterozygous, and heterozygous by heterozygous progeny, and replacing these allele calls with codes for the mapping software. So I'll go through an example of that. And just to give you guys a heads up that this is a diploid population as an example. So first, it arrives as a text file, and these arrive as allele calls, and they were scored in using Genome Studio by the Douches lab. And you just simply move them to Excel by copying and pasting, and they'll just automatically separate in Excel, or should be able to in Excel. The next step is to add the annotation to the genotype file. So here on the right-hand side, I have the genotype file, and I've taken that annotation file and I've opened it in the same screen, just on the left-hand side. And with this annotation file, I like to keep, it's only my own personal preference, but I like to keep it gray so that I know what was sourced from the annotation file, what was sourced from the genotype file that I had originally put together. So I start by inserting 5 blank rows in the genotype file, and I copy these columns A through E in the annotation file, and I paste them into the genotype file. And then just to make sure that I have the SNPs all lined up where they should be, I run the true-false formula. So here in F, I've inserted a blank column, and I do equals A2 equals G2 is a question. And then I get this series, hopefully I see true all the way down, and that tells me these SNPs are actually matched up with these SNPs. And I can actually quickly check that instead of scrolling down and looking at 8,300 SNPs by I can just do a filter in row 1 and make sure that true is my only option. If you see a false in there, that means that you have some SNPs that are not lined up with each other. So at this point, What I like to do is start recording each of the steps, and when I'm doing it, each of the steps, and take notes. So from this point on, I'll be recording how many SNPs are eliminated at each step, or any details that I think are important. So in the following, these are just some general guidelines that I discussed with the Douches Lab just to get started on working with your SNP data. It's by no means specific criteria that you need to use with your population. So next is filtering out missing SNPs for both parents. So I start by setting my filter, and then I go over to parent 1, and I deselect -- which is the missing data. And then I go over to parent 2 in the same filter, and I deselect that -- for parent 2. And then in the bottom left-hand corner, this tells me how many SNPs I have left. So I go back to my steps, notes, and in step 3, filter out the missing data for both parents. I said how many I kept out of how many originally. I then copy and paste this data into a new tab to start step 4. Step 4 is filtering out bad and questionable SNPs. So I go over to my selection here, and I deselect bad under the comments column, and then I deselect questionable, and then again I note how many SNPs I have left out of how many, and I note this in the steps section. And I copy and paste the filter data into a new tab. And then I want to filter out greater than 2 hits. So then I go down here and I deselect greater than 2 hits under PM chromosome. And then again, note how many SNPs I have left. So I'm recording the number of SNPs retained at this step. Copy and pasting the filter data to a new step, which is step 6. And I start this by inserting a blank column with a header, and the purpose of this is to filter out SNPs with missing data for greater than 10 progeny. So I'll show you guys a easy command in Excel that will allow you to look for that. So under H, I said number of progeny with missing data. That's what I'm looking for in this column. So I just type in equals count if. And this will ask from you a range and a criteria. So our range is all the progeny, so you highlight the progeny, and in this case, in this subset, extends from I2 to N2. And then you type in a comma and -- which represents the missing data again. You push enter and you fill in the formula for all of the SNPs that you have. And then we use this filter function again, highlighting across 1. You filter, you go over here, and you check for any SNPs that have greater than 10 progeny with missing data. So there are none in this case because our only option goes up to 6. So in this step, again, I note how many I'm keeping, even though it doesn't change. At least I remember that I checked it. So the next part is to filter, at least if you're going to be doing mapping, filters for those SNPs that are heterozygous for parent 1 and homozygous for parent 2. And so the first step is we set our filter again, go up to parent 1, and we deselect AA, CC, GG, and TT, and that leaves us with the heterozygotes. And then we go over to parent 2, and we select AA, CC, GG, and TT, so these are the only ones selected, and those are the homozygous. So here we can note how many SNPs that we'll be working with in the bottom left-hand corner. And here I'm noting that in my steps. So this slide I just wanted to point out again, if for parent 1, we should just be seeing heterozygotes, and parent 2, we want to see homozygotes. We move this into a new tab. So now when you move this into a new tab, you actually want to copy and paste it twice. So up here, I've copied and pasted it, and then you scroll down to below that data set and paste in this exact same data set. And in this bottom half, this is where we're going to do the replacement coding for what we want for your mapping software. So what we want to start doing in the line of the first SNP of the first individual is type in equals if, and you see this series of if statements. So in this if statement, we're saying that if SNP1 of individual 4-1 is like parent 1, it is to be coded as heterozygous, or LM is what our coding will be. So here's the top of the spreadsheet, and here's the bottom of the spreadsheet. The bottom of the spreadsheet is where we're writing our if statement, and the top of the spreadsheet is what's being referenced. So I1 is SNP1 of 4-1, and O2 is SNP1 of parent 1. So then I continue typing my code, and in this portion of the if statement, we're saying that if SNP1 of individual 4-1 is like parent 2, it is to be coded as homozygous or LL. So again, this is the top of the spreadsheet, and this is the bottom of the spreadsheet where we're typing in our if statement. And in the top of the spreadsheet, I2 is SNP1 of 4-1, and P2 is SNP1 of parent 2. So then at the end of the statement, we type in check, and we want check to appear if the allele call in that particular SNP of that particular individual does not match either parent. You fill in the formula for the rest of the progeny and the SNPs. And here you can see LL, LM, the dash dash, which means it was missing, and check. So now I want to check how many individuals per SNP, the SNPs are along here, do not match either parental allele call. So then I type in another COUNTIF statement, so equals COUNTIF, and my range is the progeny, and the criteria is the word check. And I fill this in all the way down. And again, using the filter function, we can see what our maximum is, and our maximum is 1. So finally, you want to copy and paste special as values into a new tab, and you're now ready to use or manipulate the file further for mapping. You continue this process to determine the homozygous by heterozygous crosses and heterozygous by heterozygous crosses, if that applies. And I'd like to thank you for listening and be happy to take questions at this time. Thank you.